Hello, everyone, virtually, and thank you for joining today's casual chit chat, is what I would call it. My name is Mehmet Berkmen, but my friends call me Memo, and I've been working together with Maria Pinilcobo, a local Spanish-born artist making bacterial art. And excuse me, I will talk about that adventure of ours today. All right, so today I want to talk to you about a casual discussion that I want to have with all of you regarding bacterial art. And our ambition with bacterial art is to bring the communities of science and arts together and to bridge the gap between bacteria and humans. So there's going to be 3 sections to this talk. First, I'm going to talk about bacteria, and then we're going to talk about how we make art from living bacteria. And then at the end, I'll talk briefly about the future of bacterial art. So why do we do bacterial art? Well, first of all, bacteria are amazing. I've always been in love with them since the day I met them, and I want to share that enthusiasm with you. And we want to bridge this gap between humans and bacteria. There's a big knowledge gap of what bacteria do and our relationship with them. And we have this artificial separation of science and arts that we think, using the universal language of art, we can bridge those gaps. So let's begin with what bacteria are. They are very, very old. And what I mean by that is our planet is about 4.5 billion years old, and it was a molten globular rock in the beginning, and it started cooling off. And as soon as the crust and water formed, soon after, very immediately, in 100 million years or so, life formed. So there's a big debate on how life formed so quickly. And some people think it came from other planets. And the evidence of bacterial life on this planet is about 3.5 billion years old in these fossilized stromatolites in Australia. And these are these fossilized bacterial structures that we can detect today. And just to give you an idea, multicellular creatures began about 650 million years ago, right around this period. And this planet has been dominated by bacteria ever since. And these multicellular creatures, Dictensonia, as they're called, are these sponge-like, soft-like things with very little structure. And if we look at these stromatolites, we can see bubbles of air where photosynthesis was just about invented, and oxygen was being produced and released into the air. And this invention of photosynthesis eventually accumulated. And once plants joined the system, then there was so much oxygen in the air that the Cambrian explosion began, and life started diversifying. And one major reason why multicellular creatures happened and lots of animals came onto the land was the oxygen that the bacteria produced resulted in the ozone layer, which then blocked the UV. And once the UV light was blocked, then animals could leave the ocean and come out into the land. Because once you came into the land before oxygen, your DNA would be damaged and you would not be able to survive. So much started thanks to the invention of photosynthesis by microbes. And looking at the oldest evidence of bacteria, they found these structures, these iron-like structures in ancient rocks that are about 3.5 to 4.3 billion years old. And these fossilized iron remnants and these cellular creatures in these rocks are very similar to the bacteria that live in deep ocean vents today. So there's evidence that life indeed began with microbes very, very long time ago. And just to give you an idea about that length of time, what you're seeing is just about, a little slice of this planet. And if we were to go down 3.5 billion years old, we'd have to go way, way deeper. An origin of life that began with microbes resulted in all the animals that we know. All the plants and animals and archaea and bacteria all derived from these early microbial creatures. And they're also huge. This is a misconception. What you're seeing here is a pinprick needle, the tip of a needle. This blunt end is actually the tip of a very sharp needle, and these yellow dots are bacteria. So they're incredibly small at a singular level, but when organized, they're one of the biggest creatures on this planet. So here are satellite images of microbial communities in the ocean, these algal blooms. Here you see the size of Ireland, tip of Italy, and here you have Norway, Denmark, and Scotland, and you can see these algal blooms, the size of countries that are visible from outer space. The only other creature that is visible from outer space are trees. So after the trees, the next thing that you could see from outer space are microbes. And they can organize into these giant country-sized structures. And they're highly diverse. What I mean by that is if we take all the DNA on this planet, on every living creature on the surface and ocean, and sequenced it, you would see that majority of the DNA and all the genetic information on this planet is stored in microbial creatures, bacteria, and very few amount of DNA is stored in eukaryotes and archaea. So the diversity of genetic information on this planet is mostly stored in bacteria. And you can see that most of the animals on this planet are microbial creatures. So if these facts of amazing features of bacteria, when you look at them by numbers, quite an interesting numbers start coming out. So for example, if we look at the mass of microbes on this planet, it is estimated, something we can never measure, but it's estimated it comes to about 10 to the 30 cells are on this planet, microbial cells. And just to compare, that is vastly much more than the stars in our galaxy or in our universe. So there are more microbes on our planet currently than all the stars we can observe in the universe. And if we dried the oceans and just weighed whichever creature is out there, 90% of the weight of biomass on this planet is microbial, and half the total planet's weight is microbial. So they are incredibly present and dominant creatures of this planet. Not surprisingly, they carry out most of the key chemical reactions of this planet too. Or almost all of the nitrogen on this planet is recycled back to its active form by bacteria. Majority of the carbon and sulfur is recycled by bacteria, and half the oxygen we breathe on this planet is produced by bacteria. So they're the key chemists and running the show and the ecosystem of this planet. And here's another way of seeing the microbial effects on our planet. What you're looking is a collection of about 20 years of collection of data where they look at production of oxygen. So you're seeing either the chlorophyll in the oceans here or land vegetation. And this is an indication of how the planet is breathing over time. And what you will see here, I hope this works. Yep, there we go. Is as you can see, the planet is breathing during the summer and exhaling during winter. And you can see this massive, massive shift in microbial blooms over summer and winter periods. And it's a cycle that this planet is doing through the actions of the bacteria. They're also one of the only creatures that I know of that's free of the sun. What I mean by that is if we have a nuclear war and we block the atmosphere of the sun's effect to our planet, all the creatures on the land will most likely die due to lack of oxygen and the dependence of everything on plants. But bacteria wouldn't care at all. Deep down under the ocean in these vents where it's super hot, bacteria thrive and live by chemically metabolizing these chemicals that are coming out of these vents. These are inorganic sulfur, nitrogen, oxygen, and other compounds coming out. And they can use those chemicals to breathe, to live on. And then other creatures will then come start feeding on the bacteria. And just around this vent, you can have a small ecosystem develop, and that's completely free of the sun. And all these abilities of bacteria that results in all their versatility makes them incredibly diverse and adapted to very extreme environments. They can live above boiling water temperatures. They can live in extremely acidic and alkaline conditions and extremely salty conditions. These conditions that no other creature on this planet can tolerate. And that makes this planet quite unsterile. They can breathe into the rocks, so they don't need air. What that means is that if wherever we look on this planet, we see microbes. So if we collect dust particles 3 kilometers up in the air that have been blown away, we will find microbes there. And it is estimated these floating microbes in the atmosphere are one of the reasons why snowflakes crystallize. So inside every snowflake, if you look at the center of the snowflake, you will find microbial creatures more often than not. If you look at the bottom of the earth, you go 5 kilometers deep in mines in South Africa, they crack rocks open there, and inside the rocks are bacteria. And wherever there's water, we expect life to form, microbial life for sure. And there is evidence that there's liquid water underneath the frozen ice of Europa, the moon of Jupiter. And we have strong suspicion that microbes are already there living. So putting together that they're diverse, they're important, that they're relevant, that makes them very important for our health. There's lots of evidence that eating dirt and ingesting microbes is very good for your health system, and that we have to be outdoors. And prisoners are forced by law to be 2.5 hours outdoors. And I'm sure, especially in the times of COVID, most of us aren't outdoors that long. So we really need to appreciate their role and their importance. And like the planets, that they're everywhere, similar, we're part of this planet, they're everywhere around us. Wherever microbes can reach, they're living on us. And it's estimated that the weight of bacteria that's on you right now is equivalent to the weight of your leg, knee down. So if you cut half your leg and hold it, that's how much bacteria is on you right now. And that ratio changes every time you go to the restroom. And one other way of looking at microbes that are on us is it's another organ. That's a common theme emerging from the microbiome community, is that it's an organ doing some special features for us. For example, without the presence of microbes, we would not be able to eat any cellulose at all. And a lot of the food we eat is processed by them, and then we eat the byproducts of their chemical reactions. So they are doing much to protect us, to make us healthy, and we co-live with them on this body of ours, just like the planet. And here's another great study conducted on the Karelia people, here shown in purple. And these people live in part of Finland and Soviet Union. And after First World War, they were split in half. One half went to Finland, the other half went to Soviet. And the Soviet half remained in a very rural setting with collecting their own water, their own milk, their own meat, their own vegetation, and having very little to do with the modernization of 20th century. While the Finnish counterpart, they joined Western civilization and joined the Western lifestyle. And people noticed that this community that was all one, genetically homogeneous community, after the splitting, showed different health symptoms. The Finnish Karelia people had a huge amount of allergies, while the Russian didn't. So some group of scientists investigated what was going on, and they found that on their skin, there was a complete different ratio of microbes on them due to their rural setting. The Karelia people in Finland had much less of these soil bacteria, as you can see in this graph. And they also had a complete different antibody repertoire against common allergies. So they concluded that interacting with microbes lowered your allergies. And it's something we're seeing in our society a lot, especially in developed societies. And there's more and more correlative evidence of the role of microbes have in our guts, beyond just digesting food, but on our mental state. Here's a very interesting study where psychologists collected the poop of a bunch of mental patients of theirs, and then gave these poop to microbiologists and asked, can you see if you can correlate anything? And without knowing the source of the fecal matter, they said, okay, this group of fecal matter has this type of bacteria, and this group of fecal matter has this. They just grouped the fecal matter into what type of bacteria they had. And it correlated very strongly with people's mental state. So those suffering from certain mental illnesses had different type of bacteria than those suffering from others. So this is not a causality, but it's a strong correlation. And then they found out that the people who are having depression happen to have bacteria rich in enzymes that produce serotonin. So there are some interesting links that are appearing. And coincidentally, I was reached out by a PhD student from the Netherlands who's working on the relationship between microbiota and mental disorders. And for her thesis, she reached out to us and asked if we could share this image with her, which was one of the works that Maria did. It's called Brain Matter, and which we happily shared with her. And in return, she will send us her final thesis, hopefully having this in her cover. There are also correlations between those that survive cancer treatments better, have different microbes than those that suffer more. And here's another interesting by a local Boston group where they looked at the microbiome of athletes. And they found that these group of athletes had more blue type of bacteria than red type of bacteria. And upon running the marathon, their microbiome structure completely changed. And they just assumed, not assumed, but they hypothesized that looking at the bacteria that survived, they make more lactate, more sugar, which enhances your performance. And since this is Boston, immediately the study resulted in a company that now sells you the poop of athletes or the bacteria that are from athletic to enrich your sugar uptake. And here's a crazy one. I actually thought it was an April Fool's joke, but it isn't. But the effect of having Christmas dinner with in-laws on gut microbiota composition. I think this is a tongue-in-cheek article. I don't think it was really meant to be that serious, but it highlights that, you know, we should not take this data that seriously, because a lot of it is correlated and not causalated. So the relationship between microbiome in our guts and our health is established, but the mechanism is not. So microbes are amazing. They have a huge role in our planet. They have a huge role on our body. They play massive roles, and they're very important in all sorts of aspects. I can go on for hours and hours describing this, but yet we, the media, the public, we don't really talk about microbes in the sense that scientists do. And I think the main reason for that is that we are very visual creatures and we don't see microbes in our daily interactions. Life is mostly invisible to us. So life on this planet occurs from the micrometer to the meter scale, a vast scale with many different organisms. But our eyes only perceive a narrow slit of that vast life forms that live on this planet. So we believe in what we see and we don't believe in what we do not see. And when we see bacteria, are usually in forms of rot, when things are rotting and when your breakfast is ruined. So our instinct of seeing bacteria is discussed, probably to protect ourselves from illnesses, but we do eat bacteria every day. It's just that we don't see it. And when we do hear bacteria in the media, it's usually in the forms of a disease, an outbreak, and never in terms of scientists found amazing positive effects of bacteria. That never makes the headlines. But if 5 people die from some listeria outbreak, that's definitely a headline. If it bleeds, it bleeds. And how would we see bacteria, right? Imagine if we could see bacteria. One of my heroes is Geordi Lafarge from Star Trek. Those old enough would know it. And he had this cool sci-fi glasses that he put on, and he could be able to see any spectrum of energy. He could make it see that he sees UVs or X-rays or particles of unknown origin, tachyons. And I always wondered if I could wear his glasses and just switch it and see microbiome everywhere. If I could just see the bacteria on our bodies, on our hands, on our faces, on air, everywhere, how would that world look like? And we and Maria did that one day. And I asked Maria if she could go ahead and kiss an agar plate just so we could see the bacteria on her plate. And she did. And it is estimated that about 50 million bacteria are exchanged in one single kiss. So this is another way of looking at a beautiful kiss. And Tasha Sturm, another bacterial artist, made this very famous, I guess, handprint that went viral, and it went all over the world. This is quite an old image. And it also shows the bacteria that are on our body all the time. And me and Maria, we wanted to replicate this many times, but we couldn't. This is the best we could ever come up with. So I actually think Tasha could have played with poop before making this image, but I'm not sure. Anyway, so that's about my quick intro to how amazing bacteria and how much I love bacteria. Now I want to briefly mention the separation that we have between science and arts and what I think is going on. And this might not be everyone's opinion. And I think 2 important events happened in our history that separated science and arts. One is the battle of Aristotle with other schools of thought. At his time, there were many schools of thought, all trying to explain what truth is. And one of the groups that he couldn't shut up were the sophists. And they were of the opinion that rhetoric and talking, you could explain things by rhetoric. They were very good at arguing. One of the things they would say to Aristotle is, okay, fine, you think this, but I don't. For example, you can think that the sun is yellow, but for me it's green. You could think that it's daytime right now, but for me it's nighttime. What are you going to do about it? And Aristotle was going crazy trying to shut them up, trying to come up with an absolute version of truth that could not be debated by anyone. And he came up with a concept that wasn't thought of before, which was matter. That everything in the world is made out of matter, and then eventually down to atoms. and that everything that is not real is not made out of matter. For example, your opinion that the sun is green is of no matter. So there's no matter that holds that opinion. So it's not relevant. Whereas the sun, if it's yellow, you can measure its light, the photons coming from it, show that it's yellow with a spectrophotometer and prove that it's yellow. And your opinion that it's green would not matter. So it was a nice philosophical trick to shut the sophist. And that went on for, till today. We all think that everything's made out of matter. And another event that happened was the Industrial Revolution. Pre-Industrial Revolution, the accolades of scientists, the accolades of our knowledge keepers were the deans, the professors, were of backgrounds of philosophy, literature, poetry. And those would be the deans of Oxford, and those would be the ones whose works of thought that everyone would respect. But coming out of the Industrial Revolution, what happened was schools were not churning out philosophers anymore, but they were churning out what came to be called scientists. The word scientist was first termed in late 1800s in relationship to the word artist. And what they wanted was students who came and who could fill in their factories so that they could measure Reynolds number, temperature, measure the pressure, the chemical reactions, the electricity. They did not need philosophers anymore. So both these events, the concept that everything's made out of matter and the demand of the Industrial Revolution for scientists, resulted in what I think is a little crack in our understanding of our truth. And that little crack today, over 200 years of constantly believing in these two philosophies, is now a Grand Canyon. So we have in a campus, in a university campus, on one end, School of Arts, and on the other end of the campus, we have School of Sciences, Natural Sciences, and they hardly interact, they hardly talk. They actually sometimes look at each other as enemies of truth. And when they do interact, it's usually superficial, not a deep collaborative interaction. It's usually a project interaction or a visit from one of the others, but it really isn't a common way. And I think our separation of art and science is cultural based, is historical, and is not based on truth. And so there's a lot we can and should do to bridge this artificial gap. Art is a great way of describing the truth, as valid as science, in my opinion. We started doing art as soon as we probably started thinking and capable of. And so here are some of the oldest works of art. And again, we use art to describe truth. So here's perhaps a hunting scene on our left, and here's another work of art on our right, describing what a protein looks like. Now, these proteins, they don't have arrows and loops like this, but they really describe the truth really well. And when I looked into why we represent proteins in this artistic manner, I found out the work of Jane Richardson is the reason. She's a great protein scientist. And before her time, proteins were represented by these wire-like structures showing the loops and strands. And it was very difficult to understand what was going on or going inside the protein, right? But proteins have certain chemical structures that are repetitive. And so what Jane did was standardize that kind of structures into an artistic form of helices and strands. So proteins are represented as helices and as strands. And she was inspired to come up with the concept after seeing Escher's peeling face work. So this is how we now draw proteins in a very artistic way that most scientists, don't bat an eye. They think that's quite fine to represent truth in an artistic way. And yet the gap remains. And a lot of scientists still remain questionable to using art to describe truth. This was an article I came across in a very famous journal, Cell, and the title was "Artists Create Puzzles, Scientists Solve Them." And I was baffled by this title, so I read it, and what the author was claiming is that he was using this very famous painting from Diego Velázquez, and in this painting, it's a big mystery for the artistic world. Here you see Diego right here, blown up here, looking at us, viewer, and one would imagine that this big canvas is what he's drawing on, looking at us, so perhaps we're the subject. And if we're the subject, then here in the mirror, we could see the king and the queen is where we stand, so perhaps we're the king and queen and he's drawing it. But then why is the princess in such an artistic pose? Perhaps he's drawing the princess and the king and queen visited, and why is the vizier, the person in charge, looking at us? So this was an example of how artists create puzzles And science, in fact, solve these kind of puzzles. And I totally disagree with that statement because more often than not, we create more puzzles than solve answers. Here's a classic example by one of my heroes, Howard Berg, who studies bacterial motility and chemical communication. And here in this paper, what he did is he took bacteria and just spotted on in the middle. That's it. Take bacteria, spot it on in the middle, or another type of bacteria, spot it on in the middle. And by manipulating the chemicals in the media, the bacteria naturally would form these amazing patterns without any human manipulation at all. So this was immediately a puzzle. And these kind of puzzles that we create in the lab are not solved immediately, sometimes never solved, sometimes solved in decades or centuries. So scientists, just like artists, are in the business of looking at the planet, trying to understand what's going on and trying to represent what they understand in a rational manner. So there's a lot of commonality between art and science, and this separation that they're distinct and very separate is completely false, in my opinion, and we need to bridge that gap. So how do we bridge that gap? Well, I realized when I first started doing bacterial arts in my lab that you cannot do anything properly as a hobby. You really have to engage yourself if you want to do anything well. And I wanted to do superb bacterial art, not just doodles. And I realized that to get to that level I wanted, I would need to work 8 hours a day, and I just did not have that bandwidth. So I looked for an artist collaborator to work with me. And I bumped into Maria, and luckily we've been working together for almost 10 years now. And I want to briefly introduce Maria to you. So Maria was born in San Vicente della Barquia in this northern town of Cantabria. It's a beautiful little town. I really want to go there one day, surrounded by mountains and oceans and rivers. So as a young child, Maria was highly, highly influenced by nature and her surroundings. She noticed all these wonderful patterns that are happening all the time. She saw that leaves have these amazing intricate structures, and sometimes just walking on the beach, you could find a piece of rock that has this algae growing on it in the shape of a heart. And she would see shapes and patterns that are dynamic and changing. And it really inspired her to become an artist. She went to the University of Madrid to learn fine arts. And there she had exhibitions doing artworks that were all made from natural products. Here you see it made from hemp, an insect cocoon that you could actually go into it and feel like an insect. She had exhibits using all sorts of organic matter in nature. So she was very much involved and inspired by nature. And she was using things she could naturally find, make herself with her own hand. And she came to America about 2010. And soon after she came to America, I was at a restaurant in Beverly, Massachusetts called Soma. And there, this is after me dabbling with bacterial art and realizing I need an artist collaborator. I saw these works of art on the walls of the restaurant and was immediately inspired and fell in love because it was exactly the type of style that I'm looking for. You really can't do intricate designs with bacteria, so you need some sort of loose shapes. And the shapes that I saw in this artwork were of organic and nature, so I immediately wanted to know who did it. These were these wood carved prints, I guess. You carve the wood and then use the carved wood to make these prints. You can see that they have a lot of natural shapes. These are the kind of structures that I naturally see under the microscope. And so when I realized it was Maria doing this work, I said, would you mind coming to my lab? I want to show you something. A week later, she joined the lab. I brought her to NEB, New England Biolabs. It's a biotech company where I work at. And this company really promotes and supports art. It's a very beautiful building with lots of artwork. And when she came to visit me in the lab, I showed her bacteria, and you could see that when you look under a microscope, the patterns that you see here are very similar to her artwork. And she immediately fell in love with bacteria. And I said, hey, would you like to work with me? And she said, yes. And that was 9 years ago. And she volunteers her time, joins the lab when she can, and we do bacterial art together. So in this picture, I'm the artist and she's the scientist. Because another thing I don't like is the title artist and scientist. I think all scientists are artists, and I think all artists are scientists. So we must have a more unifying term for this. So when she joined the lab, I showed her how to manipulate bacteria, how to make her own agar plates, how to work in a sterile, safe manner. And her new studio became the lab bench. and she started learning how to use bacteria. Here are some examples of colorful bacteria. And she dropped her wood carving equipment and picked up the inoculating loop, and glass beads and Eppendorfs now are her artistic brushes and tools. If you want to know more about how to make agar art yourself, we made a video together with ASM. Please go type how to create agar art and you'll find the video and you can see how to do it yourself. So I'm going to now get into the bacterial art aspect of the talk. This is our palette now. A lot of people ask, well, what type of bacteria do you use? Well, we use something we call guests. When we do experiments, sometimes we get contaminations. We have to throw away the experiment and we don't like it. But after Maria joined the lab, we stopped using the word contamination. We now use the word guests. And these guests, if they have any special color or feature or three-dimensional shape, then we keep them, we identify their DNA and find out if they're pathogenic or not, and we use them. Another type of bacteria we use are what we call production strains that my company has. These are bacteria that have enzymes in them that my company uses to purify those enzymes and cells. And these bacteria naturally have really interesting colors. This one, for example, I just have to take a little minute to explain Deinococcus, was isolated from the nuclear waste in the Canadian Shield. So Canadian nuclear reactors produce a lot of radioactive water, which they store underneath the earth. And some scientists had the crazy idea to see if anything's growing in there and found a bacteria living happily in the nuclear waste. And then they measured how resistant it is to radiation. Deinococcus is resistant to 5,000 rem of radiation. 5 rem is enough to kill a human instantly. So it is humongous amount of radiation that they're resistant to. And this planet has never seen that level of radiation. So people wondered, why are they this radio resistant? And after years and years of studying, they found out that Deinococcus is also resistant to desiccation, to drought and very dry conditions. And the mechanism of protecting your DNA against desiccation happens to also give you a resistance to radiation. We also have E. coli with chromogenic proteins. This E. coli naturally is this bland yellow color, but we can genetically engineer into these bacteria chromogenic proteins that will give various colors to them. And some of them are very beautiful that Maria uses. So this is an example of our palette. And then another question I always get is, well, why do bacteria have color? Well, it always depends on each bacteria and each color. You have to ask. There are some studies on the color and what it comes from. So from Deinococcus and Arthrobacter, I looked up, these are the chemicals that result in these colors. And a lot of these chemicals are, they have roles in protecting the bacteria against oxidative stress, or they're signaling something, or they have some secondary metabolite properties. And because the nature of color is absorption or interaction with a photon, they're also unstable, because when a photon hits the structure, it vibrates the molecule, releasing another photon, and that vibration interaction makes the molecules naturally sensitive to degradation. This is why colors fade over time. Here's another interesting, but I have to also mention, you have to remember that bacteria invented color billions of years ago, way before eyes were invented. So for many billions of years, the planet was a colorful planet where no creature could see it. So color became first, and then we had eyes. So here's another interesting study. This is a streptomyces, quite a famous bacteria for having all sorts of cool chemicals. And the scientists studying this streptomyces saw these beautiful blue colors. And just that beauty of the blue color was the reason why they studied it. There was no biological hypothesis. They just said, what is this beautiful color? And they published the paper identifying the colors, which happened to have antibiotic properties. So a study came from an aesthetic view of a bacteria, and they found out what the chemicals are, what the colors are, and have some idea of how, what their properties are too. So there's the red diffusible one that's in the agar and the blue one that's outside. And bacteria can also naturally produce amazing shapes. So you don't have to be a great artist to make art with bacteria. You can just put bacteria on a plate, let it grow, and they will make naturally these amazing shapes. You need a bit of luck and perhaps some manipulation. And for example, from a kiss of Maria's on a plate, we isolated this bacillus, which when grown for a long time, it starts making these pigments in its center. And if you just put a bunch of bacillus side by side, they start making these wonderful, wonderful shapes. But Maria just doesn't use the bacteria for their natural shapes, but she uses them to paint with them. So I want to quickly go over a couple of examples. So here are some flower-like structures that Maria made, and that's the red center bacillus that I was telling you. You can see how much the bacterial colony has to grow before the red center appears. And I asked bacillus experts to explain me what this is, and most of them, none of them knew. Here's another artwork that kind of reminds us of the Zen Japanese style of painting. Maria has a lot of interest in the sea and marine shapes, sea dollars, jellyfish, and such. And she's done many, many wonderful works with them. The brain is a theme that somehow Maria is always interested in. And here is her original wood carving drawing, and here are the bacterial art, and this is her with her daughter on the beach. So this brain image keeps on coming up. And this is another interesting work that Maria did. We bought online a mold of a brain, and we poured agar into it, and then split the agar upside down, and she then let this brain just stay outside for weeks, and we had no idea what was going on. And it started stinking the lab, and people were kind of getting upset. What is this rotten brain doing in the lab? And it, started to stink more and more, and really wonderful shapes and designs started coming out of it. And eventually, after weeks of stinking the lab, she sealed it in epoxy, and it was exhibited in a gallery. And in that exhibit, Maria came to me and said, "Hey, Memo, do you know why I did that brain?" I'm like, "No idea." It's because what she was doing was she was letting the bacteria, the microbiome exuding from our bodies in the lab, come attached to the brain. So she was trying to capture the laboratory's microbiome that's floating in the air. And so I was blown away, and I was like, wow, that's really poetic. And the weeks of suffering the stench was finally worth it. She's also done commissioned works for pharmaceutical companies. This is a company working on the microbiome. Here you see the gut, where the gut ducts, the representative bacteria here, the capillary, the villi, and the epithelial cells. And it's a wonderful piece of work. Sometimes we find dead insects lying in the garden in our campus. We give it to her, and she'll take these objects and turn them into art. Sometimes Maria just does random art where she picks pieces and just throws them and splatters them, or she just shakes the bacteria around and wonderful shapes come out of it. And sometimes these contaminations and just random bits of bacteria alone make these beautiful patterns as if it was a work of art. And sometimes just when we leave a plate outside drying and it dries and crystallizes, then we have these beautiful shapes coming out. These are the pinabacillus that come out. I think this was from the belly button of her daughter. And you can see these wonderful shapes and colors that come out. She's also worked with adding food coloring to the agar. So she would take food natural colors and add it to the agar and make these blue, red, like agar-like structures and use them. We started doing even more complicated collages where she would make these amazing drawings coming together from multiple plates. And then we started kind of getting bored of this two-dimensional flat canvas-like petri dishes. And we want to go more upwards, three-dimensional. And the idea came when I was talking to Maria, and I said, what if we could see the bacteria in our face? Similar to what I said at the earlier part of this talk. And that made us buy masks, which we then turned into molds. And she started applying these fantastic bacteria on them. And I talked about what would it look like if we could see our microbiome on our face. This is kind of a representation, a time lapse of over days and days of the bacteria growing on a mask full of agar. And Maria has bought other interesting small molds of the brain, of course, and the heart and the face. And she was manipulating them to make different structures. We even made a two-sided face for a mannequin. And this is another interesting one is my lab is the first lab in the world to make antibodies in bacteria. And the scientist in my lab was looking at the structure of antibodies. And Maria said, oh, what is that? Beautiful. So I sat down with Maria and explained to her what a protein structure is, what beta strands are, what alpha helices are. And she was like, oh, that's super interesting, walked away. And then sometime later, boom, came with this beautiful piece of work. Here you can see antibodies with metals that represent strands and helices. And sometimes she takes these colored dye agar and plugs them out with an apparatus and puts them together into a collage. And we were also asked to make a work of art, a commissioned work of art last year for Boston Bacterial Meeting. So she started working on that. This is the Boston skyline and various dyes and various colors. And our story was kind of a hobby story until the ASM's 2015 Agar Art. Me and Maria were just doing this mostly for fun. And our vision was maybe we'll have a local gallery near Ipswich where I work and invite our friends over, have bottles of wine and some cheese and call it quits. We really didn't expect or want more than that. And then in 2015, ASM started the first Agar Art Contest. We submitted our work and to our surprise, we won the first place. I think it's because we were already making bacteria for several years before that. So we had already a really good understanding while all the other people were scientists just doodling in their labs. But we also won the People's Choice with this. And that started our adventure. And that event is the reason why I'm here today, because we went viral. Overnight, we were in every newspaper. I've seen our articles in Indonesian, in National Geographic. I've seen it in many languages, many articles, many web pages. And a few months after we became viral, we got invited to give a talk at a TEDx. And this was in 2016, about a few months after we won the award. And here you see her give her first talk ever. She's never given any public presentation. But Maria's first talk ever was this TED talk while she was 6-7 months pregnant. So it was a very challenging thing for her, very stressful, but heroically she managed it. And then we got covers of prestigious journals such as Journal of Bacteriology. We were also in Ripley's Believe It or Not, one of my heroes of my childhood. And then we kind of gave a pause to ASM Agar Art. And then a few years after, we said, yeah, we should do another one. And then Maria did this fantastic piece that is one of my favorite pieces. What she did was she took a large agar and she pressed it against her breast. This is at the time when she was breastfeeding, to isolate the bacteria around that area. And there were, bacteria on her skin, but the bacteria around the nipple area were different. I would assume a different ecosystem. And there were some pink bacteria around there that she isolated. Using the pink bacteria, she drew this breast with the mammary glands underneath here. And then from her daughter's belly button, she isolated different types of bacteria and then drew this placenta uterus artwork here. And then she put a string to connect the two artworks, representing the microbial connection between the mom and the daughter, which I think is beautiful work of art. And it won the third place award for the 2018 Agar Art Contest. Here's the commissioned piece of art for the Boston Bacterial Meeting. This large, huge structure of bacillus was actually a guest in a bottle somewhere lying around. So things that are, contamination, unexpected, that ruins an experiment, in our eyes now in the lab are wonderful pieces of opportunity. But, doing all this work in a lab and posting it online won't change the world and will not bridge the gap. So we really need to engage with the public, such as today's seminar in Boss Lab. And we love giving opportunities to present our work at any opportunity. So we've had galleries a couple of times. Our work has shown in universities. We've been invited to give seminars in Stockholm. But what is really fulfilling is when we get to engage the young mind, especially middle school to high school kids, because we want to engage them young and we want to kind of lower their disgust to bacteria. This is one of our ambition. When we show pictures of bacteria, especially the kids or something, there's this ew, yuck response. And we really want to lower that. And universal language of art is a wonderful way of doing it. You don't need to be educated to appreciate art. You don't need to go to university to do art. Anyone from North to South Pole appreciates art. So once you show them art, that really opens their third eye then you could get engaged with them, make them ask questions, isolate bacteria from their skin and their bodies. And it gets to be really fun. We've also been, our work was shown in a big exhibit in Liberty Science Center called Microbes Rule. Here you can see it. Just recently, here's another development. 2 weeks ago, Discovery Channel came to our house. They're making a documentary called Pandemic. And the documentary is obviously going to be all doom and gloom. So to cheer it up, they asked us to be participant in this documentary to show another angle to interaction of microbes and humans, that it's not all doom and gloom, that you can do art and fun stuff with it. And I can't wait for it to come out and hopefully share it with you all. So this is kind of an example of the bacterial art we've done and how we've engaged the public, but we want to do more. We really think these are baby steps. We're at the first stage of it. And we really think we can do a lot more than what we've done now. Things that we're interested in, hopefully with COVID gone, we'll be able to engage in, is to make movies with bacteria, because bacteria are not static. They're not those pictures that you take. They're very dynamic. They move, they change, they breathe, they communicate. So we want to capture that. How do we capture that? Well, the best way is to capture them changing over time and making movies. So here are some examples of movies we made with bacteria. Here, what you're seeing is a beaker that we're looking down, and it has liquid, and I just inoculated some bacteria, and the bacteria are growing over time. And as they're growing, they're becoming more and more, the numbers are increasing and the liquid is getting less see-through. Here you could see that they're growing. I hope this is happening. Did I stop the video? Okay, what happened here? All right, we will ignore this video for now. But to make this video, we invented this chamber called Mocha. It's a box that has a few little features to keep, here you can see it, to keep the bacteria moist. And we also did another movie here that won a video award from the FASO Bioart competition called The Blooming Bacillus. Here you can see it in hours, and you can see this beautiful picture, an amazing movie, dynamic movie of the bacteria. And this is where it gets really wonderful for me, is we realize this chamber that we built here is actually quite useful to the microbial community. People who study bacteria in their interaction would like to have a chamber like this. So I decided to write a publication describing this chamber for anyone to use, right? And it went into a prestigious journal, a Journal of Bacteriology, and got accepted. And the article I wrote had zero scientific discovery. It was all about how to capture the beauty of bacteria using this chamber. And it went into a scientific journal. And when we were invited to Fountain Street Gallery for an exhibition, I decided, here's the article that came out, I decided to frame this scientific article and put it up in the gallery as a work of art. So this was a very important moment for me, where a scientific publication was in an artistic gallery as a work of art. And it's in my opinion that E equals mc squared is as beautiful as Bach's symphonies. So we really have to make the society appreciate science similar to how we appreciate art. Another collaboration I started with Jasmine at NYU, and her lab, the lab she works in, they make yeast having various different colors. And she has an apparatus called an echo printer that can print these different colored back yeast on an agar plate. And so this is a drawing that she made that she converted to a digital format and that she printed on. And I approached Jasmine and I said, that's great, but that's more of a technology, not an art. And art really has to, be creative. This printing of the stuff is more of a wonderful technology. And so I said, let's do something else. And she's like, what? And I said, let's print some of these famous pictures to see how they work. And so I sent her these 3 digital images. She lowered their resolution and printed them with yeast art. So these are actual yeast grown agar. And these are the artworks. And I said, we could do more. We could do an animation. And she's like, what do you mean by that? I said, instead of printing these static images, why don't we print something such as God and Adam in the Sistine Chapel? But I said, men are not popular these days. Let's switch it to Eve. And I said, why don't we print a plate where God and Eve's hands are there, and then we print another one that's coming closer, another one, and then we stitch them together to work to make the world's first microbial animation. And we did it, and it took us a long time. And the previous video didn't work, so I'm not sure if this one works too. That's a shame. Here we go. So what you're seeing is the world's first microbial art. It was made by about, 4, 5, 6 plates. It took her a week to print these bacteria. So this was a very extensive work just to do this 3-second animation. And we really want to one day find the time and the breadth to actually print hundreds of these and make a featured animation movie. So this is a project that someone could do anytime, just requires a lot of patience and time. We want to bring bacterial art closer to education. We want this kind of work to be in classrooms or in your basements at home. We want public to do bacterial art. So we are in the process of actually writing a protocol. We started writing the protocol right before COVID started, so we had to put a pause on the protocol aspect of it because we need access to a lab to write the protocol. But instead, and we want to also do interactions. So because when you paint with bacteria, they will change their color depending on what's neighboring. Like here, you can see that this colony here resulted in changing of the shape here, or this one that was yellow, when interacted with the red, when you streak this way, change its color, or the pink one here. So we need to know how they interact, which ones kill each other, which ones change its color, so we can have more control of it. And right now with E. Franco, a former student of Boss Lab, we've written, half-finished an article on the history of bacterial art, which I hope to publish soon. And we're in the process with another student in Holland to make a YouTube animation on bacterial art. And this is something that just started a few weeks ago. So hopefully it will be done. And these are some of the projects that I would like to work on in the coming months. Me and Maria also had ambitions to make bacterial art jewelry, pendants, and necklaces or earrings that you can hang. Again, due to COVID, all this paused. And I was talking with a company called Mini PCR to make a bacterial art kit that could be supplied to whomever wants it. But there are some kits out there, and making a kit is not that difficult. You just need some agar plates, Q-tips. So you really don't need to buy anything. You can just do it at home. But what's lacking is content. So once you doodle with bacteria, now what? So we would like to make informative modules and videos that people can watch and read and replicate and do experiments at home, not just art. I've also talked with scientists at MIT to make bacteria that interact with circuitry, but that again halted due to COVID. And these are all the things that we want to do in the future that are currently on pause. But before I kind of finish, and I have only a few slides left, I don't want to take full credit for the work. We're not the only bacterial artists today. Perhaps 10 years ago, there were very few of us, a handful of us out there. Today, there's more than a dozen, if not hundreds of bacterial artists out there. And if you type bacterial art on Google, you'll find many, many examples of it. And also, we're not the first ones to do it. The first bacterial artists, I would assume, are about 5,000 years ago. And it was found by people studying the cave art in Northwest America, I believe in Washington State or British Columbia, somewhere there. There are these wonderful cave arts, about 5,000 years old. And these cave arts have these brilliant red ochre colors that are still quite red today. And these scientists were wondering, how come the red color remained this long exposed to the environment? So they took bits of that red color and looked under a microscope to understand what kind of crystals they were using. And to their surprise, they found these sheaths. I don't know if you can see them here, these tube-like sheaths zoomed in here. And here's one specific one, these tube-like structures. And they were like, well, what is this? And when they asked around, it ended up being a bacteria, Leptothorax. And this bacteria lives on the mud in lake right near this cave and makes these red pigments by oxidizing iron. And what's really interesting is that if you take this red mud and heat it to this range of temperature, but not below and not above, then you fix these red bacteria to these carbonite shells. You burn out their organic matter. And these shells that have this, I'm assuming iron crust red ochre, then are permanent, and the red is very brilliant and they're very lasting. But if you go below or above it, it doesn't work. So these artists knew exactly what they were doing. They knew the temperature they had to get, and they knew the process. They might have not known that it was bacteria, but I would still say they're the first bacterial artists. The first bacterial proper artist was, surprisingly, Sir Alexander Fleming, the scientist famous for discovering antibiotics. And he was a hobby artist himself. He had these kind of drawings that he would make. And some of his patients had syphilis, were artists themselves and could not pay him. And he would ask his artist patients to give him art lessons instead of paying. And using his art lessons, he kind of tried to be a better artist. And he also took these sheets of paper that he would draw on and dip them in nutrients, soak the paper in nutrients, dry it, and then apply bacteria on this paper and let them grow. And so this is the first example of true bacterial artist. And when the Queen visited his lab, she saw it and was not amused and hurried past it. And today there's a growing community of bacterial artists. A lot of them happen to be scientists. I think that's mostly due to access to lab equipment for manipulating bacteria, but also scientists naturally tend to have an artistic eye when they play with their experiments. If you talk to scientists, majority of them will say, yes, we are artists and what I do is beautiful. But there is a lot of artists who collaborate with scientists to make art, and very few of them have been like us, me and Maria, where we've been making just bacterial art for about the last 10 years. And I want to kind of close off with the famous words of Thomas Steitz. I was actually flying to University of Kentucky to give a talk on bacterial art. And on my flight, I read that he had passed away. So I was very sad. He's discovered the ribosome structure and is very famous. And in this article, he said that I could do music as a hobby, but I could not do science as a hobby. And this was very sentimental to me because when I was 16, I was a pretty good artist. I still am. I draw really well. And I love biology, and I had to pick one or the other. And I picked science because I know I have to pay my rent, and paying my rent doing art would be much more harder. But it was a forced divorce. So that's why I do bacterial art as a hobby and science professionally. And this is because I think, just like Thomas says, it's not just being a great scientist, but it's being a great artist that helps us progress science and bridge the gap. So we have to be aware of these two worlds. We have to be aware of microbes and what they do. We need to participate in events which have science and arts together. And hopefully these kind of events and these schools and bringing this knowledge to classrooms and to our living rooms will slowly bridge this gap. And eventually we'll get more comfortable with microbes and what they do in our body and to our planet. And with that, I'm gonna thank you for your attention. If you have any questions, I'm more than happy to take them now. And thank you for your attention.
Memo, thank you for that very inspiring talk. And the collaboration between you and Maria is absolutely beautiful. I have a quick question. Memo, do you know, so a lot of the art that you create with Maria, do you guys know the strain of all of that? Or is some of that bacteria unknown to you?
Yeah, I mean, some of them begins unknown, but we try not to work with the unknown ones just for safety reasons. Of the millions and millions of bacteria that are out there, only 70 are pathogenic to humans. So the likelihood of a pathogenic bacteria is very unlikely. That said, just for clear conscience, we sequence the 16S RNA of the bacteria we play with just to make sure they're not pathogenic. If you're gonna isolate your own bacteria, my only suggestion is to stay away from human fecal matter for sure, and human sources. Like, try not to work with bacteria that came from your spit or other sources of waste. Try to work with bacteria that are from soil. For example, I tell my children if there's a rotten vegetable that they don't like, oh, I'm not gonna eat it. I'm like, why? The bacteria that are rotting that vegetable cannot rot you, right? So they're specialized for food. We need specialized bacteria that live in our body to hurt us. So caution is what I would say.
And Julia has a question in the chat. Have you ever worked with bacteria on dyeing textiles?
Well, I don't know if this is the same Julia that I was talking to a few days ago. I forgot their name, but there were 3 young people that I just talked to through BOSLab just last week about using bacteria in textiles. And they approached Wendy, and then Wendy connected them to me. So we just had a very casual discussion about what we can do. And I said to them, why don't you wait for today's seminar, and then we'll go from there. And what I did in the meantime is I looked online, and yes, bacteria have been used in textiles a lot, but not in an industrial scale, more either for art or for mini projects. And there's much we can do. And I'm especially excited about bacteria that interact with your humans. So for example, with your sweat, and change color depending on your health status. So that'd be kind of fun things to do. So I haven't done anything, but I'm in the discussion with 3 young enthusiastic artists.
I guess I also have a question, Memo. So for the bacteria that produce the color, is it mainly non-pathogenic or pathogenic strains, or is it mixed across, or?
Well, there are very few pathogenic bacteria, right? So I would say 99% of the bacteria that have color are non-pathogenic, and pathogenicity have nothing to do with color. Color is just a byproduct of a secondary metabolite. Pathogenicity is usually enzymes that damage us. So, they go into our cells and destroy cells, and not necessarily chemicals that have color.
I don't want to take up
please go ahead.
all the question time. So how long does it take for Maria's artwork from the time that she starts to put, sort of loop to agar to when is it days or weeks that this sort of?
Well, it depends. The simple ones, she just manipulates them, and then we wait a few days for them to grow and done, right?
Yeah.
But those really intricate ones that you could see are, quite a lot. Those are weeks. Those are weeks, not because it takes weeks to make an artwork. It's just the process is very tedious in the sense that she, draws the bacteria on it and realizes, okay, this is not working. This is getting too big, too fast. And that she really needs to understand the bacteria. The slow growers are put first. Some grow at very cold temperatures, some grow at high temperatures. So she needs to manipulate the bacteria. And then let's say after weeks of trying out, she got the final piece. Now, if you want to seal it in epoxy, she wants to pull that agar out of the petri dish. It cracks, it breaks, start again, right? So those really award-winning, amazing ones, I would say are weeks because she works on them so hard. But you could do one and, you could draw something or just kiss a bacteria or just put a key on it, leave the bacteria from the key come out, so it all depends.
Yeah.
What I've seen over the years is that a lot of people, amateur, not advanced knowledge like Maria, if you give them an agar, they'll just do very simple designs like a smiley face or their name or something like that. And for them to draw Maria's level is quite intimidating. They realize after well, mine looks like crap. How does why does hers look great? And then they conclude that I'm not a great artist. It's not being a great artist. It's dedicating your time to understanding how they work. I would assume, I'm going to guess, about 1.5 to 2 years, it took Maria to be fully comfortable with her work. So it's something that lots of trial and error, and patience is required.
Yeah, they're stunning and just absolutely amazing. Lada, I'm going to unmute you. Lada has a question.
Hi, Mehmet, thank you so much for the inspiring talk. It was it was really a lot of pleasure to hear it. Yeah, so the question I have, so you actually mentioned the, actually let me move probably. You mentioned that the hunters-gatherers were one of the first artists, and that they used actually bacteria to achieve this nice red color. So my question is, so you also mentioned that actually to fix this color, they had to get the temperature to be from 700 to 850, something very, very high. Right. I was just wondering at that time, how, and if that's not known, that's fine. But I was just curious if you have a comment about how they might have achieved such a temperature.
I have guesses. I mean, natural fire is about 450 degrees. So getting 300 degrees more just requires a lot of air, right? And coal-like source of heat. Wood might not be good. So using coal, a lot of air. If you just put, coal-like structure in a confined oven-like thing and blow lots of air into it, easily you can get super hot degrees and melt iron, right? The Iron Age began with people not much more advanced than cavemen. So if we can melt iron, I think achieving these temperatures is not that hard, is my guess. But they really must have like experimented, because I read that paper. In that paper, they have movies where they took this bacteria, they put it on a wire mesh, they're, with a microscope, they're looking at it, and you see them, they don't do anything, and you see the temperature, and as soon as it hits 750, you see them change shape, and these rods happen, and as soon as it goes past 850, it disintegrates and breaks apart. So, just having fire will not do it. They really must have been bacterial artists, playing with the mud, burning it, testing it.
Memo, we have a question from Sylvie.
Hi, yeah. Hi, Mehmet. Thank you so much. That was truly beautiful and amazing. I have two questions. The first one is like how NEB reacted when you told them that you wanted to have this project. Were they favorable, or like, oh, what this guy wants to do already?
Well, I didn't really ask for permission,
Okay.
I didn't ask anyone. I didn't go upper management, can we do this? I just said to Maria, can you join? And she did. Then I made Maria go to a safety course in our company. So she knew what the safety is. We make sure that she doesn't interact with anything dangerous in the lab. We kind of keep her isolated from, luckily there's nothing dangerous in my lab. I don't have any, but there's acid and acid solutions. But over the years, she's as adept. So when I get a new undergrad student, new undergrad is less experienced than Maria in the lab. So over there, she got very experienced with the lab. So she really understands lab procedure. And I just, Maria gave a talk at NEB. When we were giving a TEDx talk, I made her give a practice talk a week before at NEB. I've announced to NEB a couple of times that what we do, but I try to keep our work, separate from my professional work. So when I go to lab, I work 8 hours on my professional research. And when Maria walks in, we chit chat about projects, we talk about here and there, but we don't work, like 8 hours a day. She walks in and works for like 4 or 5 hours solid. And usually our interaction with Maria is very collaborative. She has an idea, I give the idea back to her, say, how about this? She's like that. And I say, oh no, you can't do that, it won't work. And I say something and she comes up with a better version. So it's a very collaborative idea. She's very independent now, she really doesn't need me, but we really like bouncing ideas back and forth. For example, just as a recent example, when the Discovery Channel came here, we said we should do an artwork for that. And what should we do? What should we do? I said, oh, planet with COVID-like spikes, right? And then she went and she got the structure of the spikes and she made the artwork. And I said, well, the spikes are super realistic. They have the trimers and the binding. And I said, most people don't understand the trimer protein complex. Let's just make it red bulbs that you see in media. So that's the kind of interaction we have where she does something and I say no, and I say something and she says no or yes. And before you know it, a work of art is out there. It's a very 50-50 like process, although 99.9% of the work is done by her.
That's great. Thanks. And the second question is like more technical. Did you ever think about doing, about engineering a strain that makes all the color? So like a rainbow producer or something that will make different colors based on like the nutrient or something.
Wow, that's beautiful and tough. Because, if you think of a bacteria, let's say it makes 3 different colors, right?
Yeah.
Those chemicals will be swimming around inside a bacteria and you will not see rainbow, right?
Yeah.
But they naturally can make rainbow shapes in the sense that they send chemicals to each other to see how many bacteria are there. If there's a lot of them, that means there's not much food. If there's few of them, that means there's more food, right? So they always want to know how much bacteria there are. And these communications can be genetically built into a circuitry where you just put a bacteria on a plate, and I've seen examples of it, and they will make zebra-like pattern, black and white. So they will turn on a color and turn off a color, but as groups, like these will make a black, and the next lane will be all white, next will be black, and they'll make these beautiful patterns. So making patterns is possible, but rainbow, well.
So
So you'd have to link that communication, that chemical communication, turn on red, turn off red.
This is what I'm thinking. This is what I'm thinking about.
There are circuits, genetic circuits that have this already published, so you can already do this. And you could put, for example, a chemical, an inducer on one side of the plate with a linear concentration. And as the concentration, they'll make different types of color. I mean, these are the kind of things I'd love to work on, merging of science and art. But then you're talking about, you know, hours and months of work.
Yeah, especially like, is it even possible to make this strain like, because like, you know, if you produce this much things which are not like endogenous, I don't know if the bacteria will make it through.
Yeah, I mean, just this is the exact boundary between science and art. You really need to be a good molecular biologist to be able to do something like that. And that kind of molecular biology knowledge is resulting in art, not in a publication.
Mm-hmm.
So who would spend, you know, years learning molecular biology just to make bacteria? I mean, that's what I would love to do if I had the time.
Sure. Thanks. Thank you.
I think there's a question by Lada.
Yeah, I just had also like one question along a different line. So when you showed this plate where you basically were having different strains, if I remember correctly, of different colors, and then one line of one strain was like merging into another line, and you were saying that like, oh, look how how interesting the interaction is, because like the line up top kind of like influences the line in the bottom. And I was wondering, like, actually, did you explore any of these effects from the scientific angle? And and you were when you were given an introduction, you were talking mainly about different bacteria and its role, like for the world. And what is your scientific focus in NAB, if you can tell a few words about that?
Sure. Regarding your first question about the interaction, we made that image hoping to start studying it, and then COVID happened, right? So we have that one image, right? We really want to, like, we didn't even know how to study interaction. Do we do those stripes crossing, or do we do spots of agar, of bacteria, and then you see in the zone where they interact if they change? So we were going to manipulate different methods to understand interaction, pause COVID. So I really have no more knowledge than that. I do not have a scientific interest in what's happening there. I don't have a scientific interest. This is more for understanding how bacteria behave on an agar plate to make art, right? So we were gonna remember that article, we're gonna do how to make bacterial art in your home. So for that article, we needed a section called interactions, and we were gonna work on it. Even Boss Lab was gonna work on this, but COVID happened. So that put us stop. Regarding my personal scientific interest in NAB is, I'm a bacterial geneticist by training, and my company makes over 550 different proteins from all domains of life. And we make it basically all in this one bacteria called E. coli. And if you look at what types of E. coli there are out there, there's like a couple, 2, 3, 4, 5, and they're all historical hand-me-downs from various labs. We don't have bacterial strains specifically designed for protein expression. So that's where my lab's research is, to genetically engineer novel types of bacteria so that we can expand our protein expression capacity. So I sometimes jokingly say I play God with bacteria. I screw around with their genes. I don't know what I'm doing, you know, mix and match and make Frankenstein bacteria, and hopefully one of them will not kill the village.
Right. And another thing that I was thinking about when you were showing again the interaction slide, or as a plate, that actually a lot of like that bacteria actually can exchange genetic material by themselves using like different means, just pieces of DNA, or it could be whole plasmid, or it could be smaller piece, and everything. So I was just curious also, but I understand this is like a whole different topic. So, but
that's a great idea. You know, you have a bacillus that makes red dye, and then you conjugate, and it gives the information to another, and now the other one starts making red dye. Lada, you want to have time to join my lab?
Well, I wish I had. But it was a pleasure to just see the whole like breadth of work and all the possible different angles one can look at it. It's amazing.
Yeah, there's much we can do.
Yeah. Thank you so much again.
Thank you for your interest and your kind words.
Anybody else have a question for Memo? Memo, thank you. This has been like truly inspiring. And if you too have been inspired to create AGA art, this is my plug for Boss Lab. We are doing some AGA art classes that you can sign up for on Eventbrite. You can also find the link on our meetup page. You can come to this space. We are not mixing people of the general public, so we're asking you to come as a quarantine pod, a group of people that already are cohabitating together and staying safe. So check us out on our Eventbrite, Boss Lab at Eventbrite. And any other questions or comments or feelings?
If you have questions later on, you can always reach me through email.
Yes, you could reach out to, you can also reach out on Meetup, and I can connect you with Memo too. So, and we're always looking for collaborations, both groups. So if you have a great idea and you want to try something out, just reach out and contact us.
Thank you very much, Wendy, for organizing this.
Yeah, no, thank you guys. We really appreciate you.